So good morning, everyone, and welcome to the second day of All India AIIM Meet 2020. And today we also have a schedule packed with great speakers and events. So make sure you properly check out the schedule on the Slack channel. So coming to our first speaker of the day, it's Dr. Sumit Biswas. He is an associate professor at BITS Solani KK Birla Goa campus, and his working areas include the molecular and structural mechanisms of biofilm, therapeutic biology, protein machinery of Vibrio cholerae and Staphylococcus stages, and the bioinformatics of protein nucleic acid interaction. We'll now open the floor to Dr. Sunil. Welcome, sir.
Thank you, Gaurav. Thank you, all the organizers, and a very good morning to all of you. So I am very sure this is a very fruitful All India AIIM meeting that's being organized, and I hope I can contribute to a part of it. So since I've already been introduced, let me go straight to my presentation. I hope you can all see my screen. Please give me anyone can unmute and
Yes,
then.
sir. Yes, sir. We
Okay,
can see
okay,
it.
okay. Thank you so much. So basically, our lab, the Vista lab, was started with the aim at looking at Vibrio and different structural aspects of proteins from Vibrio. So later on, we migrated, and we are primarily more focused right now on the biofilm formation aspects, not just of Vibrio but other biofilm forming organisms as well. So today, what we are going to do is I am going to just show you a small glimpse of what we have done regarding Vibrio biofilms, and I will try to make the talk as interactive as possible, and I will try to focus more on showing how you can hit roadblocks and how you can avoid those roadblocks or mount those roadblocks and go ahead with your experiments. So as I have titled this talk, "A Needle in a Haystack." So the haystack is basically the mass of or the jamboree of amino acids that we have in this particular protein that I am going to show to you. And we will also show how this protein was instrumental in the formation as well as the mutagenesis of that protein resulted in the loss of the biofilm. So the basic outline of this thing is going to be mostly a synthetic design of the protein, and in parts where experimental designs failed, I will try to show how we could do some sort of modeling experiments to circumvent the issue. So if I am going to start at the beginning, I will have to start with our protagonist, that is the cholera disease. Since we are sitting in a pandemic right now, and we are all worried about what to do about the pandemic, so let me report to you that this is a pandemic which has been going on, not a pandemic, a series of pandemics. Currently, we are in the 7th pandemic of Vibrio cholerae, and this pandemics have started way back since the last maybe 4 centuries ago. So this current pandemic started around somewhere in the 1960s, and though we are all not very bothered about cholera or Vibrio cholerae in many countries, however, the situation isn't the same. This pandemic keeps on continuing in several countries across the globe. This is a little dated map because WHO hasn't come up with anything after 2015. So if you can see in this map of 2015, you can see that there have been countries reporting cholera in 2015 as well, and there have been countries reporting cholera ever since. Now, if I go to a more recent map, the one which was published in 2018, I'm basing myself entirely on the data that I get from World Health Organization. I can see that there are extensive periods or extensive parts of the world where we still have the pandemic continuing, notably in countries where you do not have the top of the level sanitation and other measures. So this latest pandemic is mostly prevalent in Africa and some parts of the Horn of Saudi Arabia and in Central America as well. And if you notice the case fatality rates, in some cases the case fatality rates, that's the CFR, indicated by the radius of the circles in these countries. You can see that the case fatality rates in some cases are quite high and have been much more than what the current pandemic of the COVID-19 has showed us. So all this beating about the bush is nothing but to highlight to you a fact that how an organism which has not been cured as of now, nor been vaccinated as of yet can keep on in a continuing fashion as a pandemic, and we can still have measures to sustain, or so not sustain but to contain those effects of the pandemic. So right now, we can see that the cholera pandemic is something which is a global aspect. And if you can see all the continents which are coming into the pictures, it's not that it has gone away ever. It has remained in it's remained in the population ever since it started in the 1600s. And you can see new domains, new territories being added in years. And that is because of the fact that the bug or the bacteria that we are dealing with is a super bug right now, and we will go through that story in a short while from now. Now, the problem with this thing is there are no real vaccines available to counteract the measures of this pandemic. Now, what has happened is that some years back it was launched. This was is a killed attenuated vaccine, and what people had done is they had taken these things. But however, not only was the vaccine hypersensitive to a number of people, but it really didn't have a long-term effect, in the sense that it lasted only maybe 2 to 3 months after the vaccination, and it was not continued hence after. And WHO stopped the use of this vaccine altogether. So you have a situation where you have a continuing pandemic and you don't have any real vaccine. And as I told you, these are organisms which are super smart. So primarily Vibrio cholerae or bugs were basically of two lineages. One was the classic Vibrio cholerae, which originated somewhere in the subcontinent along the banks of the river Ganga. And of course, the second one was the a little different Eltor. Eltor is the name of a well in upper part of Saharan Africa. And what happened in the first decade of this century is that the Eltor and the classical type of Vibrio cholerae, they together merged and created a new form which was able to imbibe the best features, not in terms of human beings but in terms of infectivity. This was able to imbibe the best features of both Eltor and Vibrio cholerae, and resulted in a new sort of biotype which had severe implications in terms of infectivity, as was highlighted in the outbreak of the pandemic in Bangladesh in 2008 or 9, I guess. Now, how does Vibrio cholerae maintain its presence in our environment and still continue to be a pandemic causing organism over the years? Now, herein, I will ask Basha to please circulate the poll, because this is a bug which you can very well see has not been counteracted by vaccines or by any other measures, but to a great extent we have been able to help create awareness about it by following suitable hygiene measures and by following certain good practices. So that's a way we have shown in the past that we can handle a pandemic, and we can still have an effective way of counteracting the effects of a particular superbug. So I think the poll is live now, and you can give a quick vote on that before we move on to the next phase of this talk. Okay, I think we can move on now. So what really is the reason for Vibrio cholerae to be able to survive over the centuries and still being a potent threat to humankind? The mystery lies in the dual life cycle of this organism. So what do I mean by the dual life cycle? If I look at the life cycle of Vibrio cholerae, okay, thank you. So if I look at the life cycle of Vibrio cholerae, I have a two-phase cycle. One is the more famous infective cycle that we have inside the human host, and it is in this phase that the Vibrio cholerae releases the cholera toxin, which causes the acute diarrhea and the loss of all intestinal and body fluids, leading to the death of the individual who has been affected. But this is the more potent and the more famous form. But the trick or the mystery behind this infectivity lies in its other life cycle form, which is the aquatic biofilm form. Now, this second part of the life cycle, where the Vibrio cholerae does not move, rather it's a sessile organism and resides as clusters of individuals in a, in a static form known as the biofilm, which is very much prevalent in an aquatic body, is the one which gives it the potency to create havoc once it reaches the human host. So in this aquatic biofilm form, what it does, it comes together on some sort of substrate or some sort of a planktonic medium and forms a dense biofilm by secreting some exopolysaccharides and some other nucleic acid and other components. In this biofilm form, this cholera the sessile cholera organisms are not susceptible to the action of antibiotics or to chlorine or any other thing that you use to purify water. So the result that happens is a saline water body that you might be looking at will be harboring a layer of biofilm of the Vibrio cholerae, which, when they will have the trigger, will try to again come back to the infective cycle in a human host. So our thought process was, how is it possible, or whether is it at all possible, to somehow disrupt the biofilm formation of the Vibrio cholerae and arrest the can I say the transformation to the infective cycle by doing something about its biofilm forming mechanism. So in order to look for that, what we did was, uh, I'm sorry. So in order to look for that, what we did was we tried looking at a family of proteins, which when we started were known as the proteins with domain of unknown function. So the trademarks of these proteins were, they had, if you can follow my cursor, they had a signature GGDAF domain, or even you can call that a GGDF sequence domain in their primary amino acid sequence. Now, what these GGDF domain signature proteins can do is that they create a molecule of cyclic di-GMP utilizing two molecules of GTP, which they sequester from the cellular pool. Now, what the C-GMP does is, now it has been extensively studied, and almost all eubacterial and other bacterial system, even some archibacterial and lower eukaryotes, have been able to show this thing. The cyclic di-GMP or C-GMP is the basic or the most potent secondary signaling messenger that exists in the bacterial world. Now, what cyclic di-GMP does is it can trigger the release of many toxic factors. It can also trigger the release of the factors which are responsible for causing the biofilm in Vibrio cholerae. Now, C-GMP is not a molecule which remains under a single pathway, that is not synthesized alone. It can be broken down as well using a separate set of proteins which are known as EAL domain proteins, and the cell uses EAL to convert cyclic di-GMP to two molecules of GMP when the need does not arise to have more C-GMP. So for this mechanism to function, you need to have some sort of enzymes which will be able to convert GTP to cyclic di-GMP. And herein came the role of the GGDF domain proteins, which are now known as the diguanylate cyclases. And these are distributed, as I said, over all the domains of the eubacterial system, and even now have been found to be present in some lower eukaryotes and other the archaeal domain as well. Now, what we did was we targeted a cyclic di-GMP, or rather a GGDF sequence containing protein from the chromosome 2 of Vibrio cholerae. Mind you, Vibrio cholerae has two chromosomes, the chromosome 1 and the chromosome 2. So what we did was, with the VCO395 underscore 300, that's the genomic annotation. What we did was, we will call it CBOX3 for the future references. So what we did was, with CBOX3, we found that there was not only this DUF1, or the domain of unknown function as it was known, or the GGDF domain in it, but it also contains something known as a secondary sensory domain, or the environmental signaling domain. Now, the thing with the environmental signaling is the fact that you can actually sense the environmental signal if you are a bacteria having a ggdf domain, and you can trigger your ggdf domain containing protein to function, creating cyclic digmp pools in your system. And these cyclic digmp pools are instrumental in causing a number of life cycle or lifestyle changes in the bacteria. So the thought process is this: if you can somehow debilitate a ggdf domain protein from functioning, would you have a system where the bacteria would not be able to perform the activities that are done by the secondary signaling messenger or cdigmp? In our case, this activity was more concerned with the formation of the biofilm. Now, if I claim annotated protein from a database to be a protein of function, nobody was going to believe me. So what we did was we tried to first find out a method to prove that our enzyme or the protein that we are looking at was a diguanylate cyclase, or it can synthesize cyclic digmp. So what we did for this was we tried to form an assay system by which we could show that the presence of the substrate, that is our protein, which we had synthesized by recombinant DNA methods in a different vector system. If the recombinant protein would have been able to convert GTP into cyclic digMP, then we could have claimed that our enzyme is indeed a diguanylate cyclase, or our protein is indeed a diguanylate cyclase. This we did with some difficulty because we missed a very significant clue, which we later on would decipher using molecular modeling. Now, I will come to that in a little later. For the moment, let's see the assay results. Here you have GTP only with a peak. This is a HPLC based assay method that we devised. And you can see CDI-GMP and GTP. Obviously, these are the markers that we employed. And you see if we could use CBOX3 in the presence of GTP alone, we could also trace the synthesis of cyclic digMP, which was produced as a result. That gave us an idea that the CBOX3 is a diagonalite cyclase. So how do you know how it works? We also went into traditional biofilm assays. So you can see over here, please focus on QRGB and CBOX3. This is a crystal violet staining method of observing biofilms, and we can see that both QRGB, which is a positive biofilm forming Vibrio cholerae, and CBOX3, they have the ability to form biofilms when grown for a period of 4 days or 7 days. So this is a complementary to that biofilm forming assay. If you form a biofilm, you are not supposed to move away from your place of inoculation. So I will ask you to focus currently on the AC3 part of the picture, which depicts CBOX3. So what we did was we stabbed agar culture with CBOX3, and if you can see that CBOX3, which is supposed to form biofilm, means it does not have motility, has actually not been able to move away from its stab line, whereas these are the mutants of CBOX3, which we will discuss very soon, and they have been able to migrate from their stab line. So these are the two factors which prove to us that the CBOX3 is a biofilm forming diagonalate cyclase. Now, even then we were not happy, so we came up with a new method of looking at biofilms, and we investigated biofilms under the scanning electron microscope, which gave us this dense picture. You can see the Vibrio inside the dense matrix of biofilms, which was formed in the colonies we had looked at. So when we knew that CBOX3 is a biofilm, our next target was to find out whether we could use the CBOX3 to see how its structure looks like. Unfortunately, we could never arrive at a crystal of the CBOX3. We tried modeling CBOX3 with respect to some similar homologs from other bacterial systems, but the best sequence homology was close to 30%, and we were not very sure of the models. We will come to the modeling part in a few slides from now. So what we did was we looked into CBOX3 structure, or we looked into CBOX3's domains, and we found that CBOX3 had a flexible region which prevented it from crystallizing altogether. Now we figured out what this flexible region would be from doing partial proteolysis and subsequent sequencing of those portions using a mass spectrometer. And we found that the primary N-terminal region of CBOX3, the region which I have highlighted in blue in this picture, in the sequence file, is the one which is responsible for the flexibility of CBOX3 and causes some sort of obstruction to the process of crystallization. So the next part we have to think of was to go for truncates of CBOX3 because we were not being able to crystallize the entire protein in its own. So if I create a truncate, I must be aware that I should have the same activity in the truncate as the original protein. That is, I should be able to display diagonal edge cyclase activity in the truncates. So when we looked at modeling or designing the truncates, we found that there were a few regions which we could successfully take out yet maintain the activity of CBOX3. And these regions we figured out stretched to around 160 amino acids from the N terminal end. So what we did was we created a series of truncates, and from this series of truncates we checked for activity of the protein, and we also checked for whether they were able to crystallize under limited crystallization conditions. And finally we came to some truncates, that is truncate number 161 to 321. I forgot to tell you that the CBOX3 is a 321 amino acid protein in its entirety. So the 161 to 321 and the 164 to 321 truncates were able to have a soluble protein expression, could show the diagonal cyclic cyclase activity, and were also able to crystallize. So to check for the activity of these truncates, we also undertook the same experiments we had done before. We measured the biofilm formation and the non-migration from the stag line, from the stab line, with the aim of verifying whether these truncates are really active diagonal cyclases or not. So these truncates of 160 to 321 and 164 to 321 we demarcated as CBOX31 and CBOX32. Now before going ahead with the full-fledged crystallization of these things, we also wanted to make sure whether we really had a structural basis for doing so. So we went for creating homology models of these truncates based on the template we had created. This is the homology model for the entire protein CBOX3. And what we did was we also constructed homology models for the truncated CBOX3, that's 31, and also 32. You can see the differences in the structures of the homology models. But what was good about all these things was that the dimerization domain that was present in CBOX3 wasn't really disrupted even in these homology models. So what we did was we use the crystals of CBOX3 truncates, and from these crystals of CBOX3 truncates we could get a good resolution. You can see here that the resolution was close to 2.1 or something. And what we could do was we could arrive at a structure of the CBOX3 in its truncated form. Now, when we map this structure to other structures of diagonal cyclases from other bacterial systems, we found that there was a very close conservation of the diagonal cyclase fold, which we could see in terms of the secondary structural components and the way they were arranged. So a typical diagonal cyclase folds looks like this. It's an array of beta strands forming a sheet-like structure, and the GGDF domain or the signature domain is also showing the presence of this GGDF loop, which is instrumental in binding to the GTP, as we will see very soon. Now here came the second roadblock. We could not really crystallize GGDF domain containing CBOX31 or 32 along with GTP. So that created a hindrance for us, and we could not really move ahead, though we knew that this was the GGDF domain and its conservation of architecture. We had to somehow model a GTP to come and bind to this loop and figure that out. So what we did was we used all the three homology models we had got as static receptors, and we assigned polar hydrogens, non-polar hydrogens. We merged and created a PDB-QT files. All of you who have worked with Autodoc would be aware of what is a PDB-QT file. Basically, it's nothing but a file very similar to PDB, except that you have assigned charges and you have assigned hydrogens from done by the Autodoc. So we also added Gastiger charges to these receptors and the ligands, and we utilize the function which is present in Autodoc known as AutoTors to define all the possible torsions of the rotatable bonds of the ligands and the and with the GGDF domain. Now, what we next did was we got a grid map around the receptor sites, and within this defined grid maps, we checked the dock poses of GTPs for all the three homology models with the highest binding affinity. So once we were done, we of course did an energy minimization using GROMAX, and then we utilized the AMBER 99SB force field, and we also added charges and stabilized that using a different software known as Chimera. So together with these things, we found that there were some GTP binding sites which were displayed in the presence of a GTP, and these sites were of course concurrent to what we had found in the structure, and these sites were very similar to what we predict as the binding sites of the GTP in the structure. Now here came in a very essential part of a clue. The the great the big peak of cyclic di-GMP that we had shown in the diagonal cyclase assay previously was not shown initially. During our modeling, we found that there was a space for a divalent cation which could be imbibed into the active site while binding to the GTP. And if this divalent cation was administered, we could have a closer packing and closer interactions happening between the GTP and the GGDF and the GGDF sequence motif. So when we added magnesium to our assay mixtures, we found that the rate of formation of cyclic di-GMP or the total amount of cyclic di-GMP produced from the cyclic di-GMP assay that we had envisaged shoot up like 5 times in the process. So this also emphasized that an actual divalent cation, that is the magnesium here that we have used, was really essential for forming the cyclic di-GMP activity of the enzyme. Now, what this did was, and it was also corroborated by other publications from other people working with diagonal cyclases, gave us a clear idea as to how the diagonal cyclase binding formation site or the active site looks like in a proper cyclic di-GMP diagonal cyclase molecule. So I will just sum up a little bit over here and tell you that the protein that we had started looking at, the CBOX3, does have a diagonal cyclase activity, which is heightened by the presence of a divalent cation and is basically a conserved architecture with other diagonal cyclases that are present in other bacterial systems as well. Now, we didn't start with the job of looking just at the structure and getting and patting our backs. The next thing we had to do was we had to synthesize mutants of Vibrio cholerae VCO395300, what we call CBOX3 in our common lab lingo. So what we did at each of the sites of the GGDEF thing, we generated a site-directed mutagenesis simply by introducing a different amino acid to this site. Of course, we went for scanning of the amino acid that we were supposed to introduce. And when we did that, we arrived at a series of mutants, which we will call CBOX 5, 6, 7, and 8, and 9, and so on. So we will not talk of all the mutants, but I will just try to show you the activities of CBOX 5, 6, and 7, which we found to have a significant activity. So what was exactly CBOX 5, 6, and 7? So if you can see here, if I had changed the second G of the GGDF sequence to R, that's what I create. If I mutate the second G, I call that CBOX 5. If I mutated the first E of the GGDF sequence, I call that CBOX 6, and so on and so forth. And if I mutated the F, then I called it CBOX 8. So if you can look at this, look at the same assays we had undertaken for the CBOX 3, you can see that CBOX 5, 6, and 7 had relatively less amount of biofilm formation as compared to CBOX 3. Similarly, if you now remember the stab line assay that we had shown, you can see that CBOX 5, 6, and 7 had much greater migration from the stab line as compared to CBOX 3. So we could safely assume that CBOX 5, 6, and 7, that is the mutants of the CBOX 3 protein with respect to the 3 central positions of the GGDEF sequence, were having an activity which showed that there has been a significant loss of the biofilm formation of CBOX3. Okay, so that's how it's summoned. So we also, as I said, undertook the same assays. You can see here that the biofilm which was formed earlier with the CBOX3 is more or less much disrupted in case of the mutants. I am showing you just the figures for CBOX5. And we also crystallized CBOX 5, 6, and 7, and we found that CBOX 5 crystals were not so handsome looking, but they had a better resolution. Actually, we could go to 1.8 or something with CBOX 6, CBOX 5, and we finally again solved the structure of CBOX 5. So when we came to the structure of CBOX 5 and compared that with the structure of CBOX 3, what did we observe? We saw that a single change, a small change that we had introduced in the CBOX 5, that is, we had changed the second G to a more bulkier amino acid, which was an arginine in the case of CBOX 5. We saw that there was a steric hindrance which was being posed to the entry of the GTP. Now, when we docked GTP to the active site of CBOX3, we saw that there was no obstacle and GTP had its permanent binding partners with the GE amino acids which were present in the CBOX3's active site. Now, what we have done with CBOX5 is we have created a mutant in which the entry of the GTP, so here is where the GTP should have entered, has been blocked due to the protrusion of a longer side chain from the GGEF signature domain. Now, what that did was that prevented the ability of the diagonalate cyclase to synthesize C-di-GMP, because if you do not allow the binding of the GTP or the docking of the GTP to this active site at all, you can actually prevent the formation of cyclic di-GMP and thereby debilitate the activity of this particular protein in general. So I am almost at the last slide and I hope I am not very much out of time. So what we have seen here you can see, if you can see the surface diagram over here. In red is what you have for CBOX3 and in cyan is what you have for CBOX5. So what CBOX5 does is simply it protrudes a bulkier side chain which prevents the activity of GTP. So what did we arrive, what did we derive from this? One, the CBOX3, which is a very important part of the biofilm forming mechanism of Vibrio cholerae, had its potency or most of its activity residing in the single GGEF domain or GGF sequence domain in its active site. When we compared the two structures, if you can see the overlaps or overlays of the structures we have, there was no significant difference in structural morphology throughout the entire protein. I mean to say that the haystack remained exactly the same. The small needle that we introduced in the form of this mutation was the one which made this protein or made this diagonalate cyclase lose its activity in entirety. So if you can introduce a needle in a haystack rather than find the needle in a haystack, you can actually cause a major biological or morphological change in a superbug like the Vibrio cholerae, which in this case was our disruption of the biofilm. Now, what's the next step ahead? We are trying to have, since we have to work with modifying a potent pathogen, there are some difficulties, but we are trying to work with imbibing these sorts of mutational, these sorts of sequence mutated forms using suicide vectors in actual Vibrio cholerae pathogenic strains, and we are trying to find out whether we can have a propagative form of these pathogens which cannot form biofilms, and the ultimate goal will be to figure out whether biofilm deficient strains of Vibrio cholerae are really able to survive in the aquatic environment, and if they're able to or whether they're able to take environmental cues and jump to a infective human form at all. So to acknowledge the people who made this possible, these are my PhD scholars, Divya, Om, and Dhiren, whose publications we had seen before. And this is Yvette at the basis synchrotron, who helped us with a collection of diffraction data. And these are some of the funders who have made this work possible. So if I think I have not overshot my time, I seek apologies if I've done that. Thank you for being a wonderful audience, and I wish I could live up to the theme and the expectations of the All India IHM meeting. Thanks a lot.
Thank you, sir. That was a great presentation. With anyone have any questions? I have one here. Should I just read it to you, sir?
Yes, please. Yes, Gaurav. Sure, certainly.
Okay. Aditi Chaudhary has asked, as Vibrio was bacteria, we were able to find its vaccine. But what about virus? Can we really find its vaccine? Is the question.
If I didn't hear the entire, if it had been, could you find what exactly?
As Vibrio was bacteria, we were able to find its vaccine. But what about virus? Can we really find its vaccine?
Can we really find that? I can't hear the last part.
Find its vaccine.
Find its, sorry,
Vaccine. Vaccine,
uh,
like the same vaccine.
vaccine. Okay, vaccine.
Yeah.
I think the question is it relevance to the present scenario, and for retroviral vectors, finding a vaccine is even more difficult job because, as I said, Vibrio being a superbug mutates and imbibes the best traits of its most infective forms. So a virus is more prone to do that. So even though we could find a vaccine for Vibrio cholerae, of course, as I told you, it's not effective and it has never been used as of now. That's why we are going into ways of disrupting it by synthetic means and manipulations. However, for a virus, it's a little more bit more difficult because because of its high mutagenesis rate, and somehow it's always easier to tamper with its life cycle rather than creating a permanent seal of a vaccine for a virus. So that's how I see this thing, and I hope it answers the question.
One more thing is the poll results. So 54% of the people actually said yes to if they think the lack of vaccine for cholera has led to better human hygiene.
Okay, so that's mostly a split of equal measures, I guess. So
Yes.
I think, yeah, I think lifestyle changes is a very critical point in targeting or trying to disrupt the life cycle of an organism which has traits of being very uncontrollable and all that. So maybe our awareness, I mean, you don't really get to hear stories of cholera infections happening even in our subcontinent as much as you used to do maybe 200 years ago. So what I feel is that there's a hygiene and proper sanitation plays a huge role in ensuring that such stubborn bugs or viruses have a way to be contained. So that's how I feel, and I guess uh,
Uh, so we have a few more questions. One is,
please.
as apart from the laboratory work, how do you plan to carry out your project practically? That is epidemiology.
Right. So as I said, after this laboratory work is done, and we are still in the process of creating mutant vibrios, which would be having this sort of a GGDF mutation in them. Now the thing is that a bacteria or a virus finds out different ways of circumventing a roadblock. I mean, they are much better at going around roadblocks than us. So what we have to do is we have to see whether we can stabilize these mutations and whether we have strains which can actually live with those mutations and not circumvent it to find another way of forming the biofilm. So once we do that, we will be trying to have these sorts of vibrios in the environment, which we will then see in terms of infectivity and spread. So that is something in the future, and that will require a lot of different biosafety containment levels, which we will try to envisage in as we go along.
Okay, so one last question because we are also short of time and Dr. Partho Roy is also here. Uh, what might the challenges in introducing and propagating these SEB5 strains in the environment be?
Okay,
Why would the Vibrio prefer to propagate this non-biofilm forming mutation?
absolutely. That is what we do not know. I mean, at present, what we have is we have a system which we think will be able to compete with other strains. But the moment you introduce into the environment, as I said, no living organism would want to adapt to a less enabled way of lifestyle. So the first challenge that we would be facing is whether these would be like adapted or these would be suited for survivability in a environment where you also have vibrios which will try to form biofilm. So how you can modulate that? You can modulate that by looking into the environment sensing domains or the environmental sensing signaling sequences that are present in its vicinity as well. So I didn't highlight anything about the PAS sensory domain that we have. And as I spoke of a truncated form where we had deleted the PAS sensory domain. So what we have to also do is we also have to look into how these things sense the environmental signals. So if these are better at sensing environmental signals, we have to create some sort of a bypass by which they don't form the biofilm, sense the environmental signal, and be better adapted than the ones which sense them less. So what we can do is we can just see and observe how we can let these two strains, that is the biofilm deficient one and the biofilm forming ones, compete in an environmental system, and then only we can figure out how we can go ahead. And that's why we will be looking into how the signatories, the environmental signature sequence are going to function in this process. So it's a long way ahead, but we are into it, and we hope that we'll have an answer to this sometime in the near future.
Okay. Thank you, sir, again for this
Yeah,
talk. And now I think we can move forward. Prajwal will take over from here.
yeah, thank you so much. Thanks. It was a beautiful platform.
Thank you, sir, for your talk.